Um, the BioCORE, if, as you recall, has these 5 categories of analysis that you look at any phenomenon through. It's like a lens through which you view things, and you kind of take notes to learn something more about the phenomenon by looking at it through these different sort of lenses. There's the structure function thing. So for any object, any sort of noun, you can look at it and see what the structure is, and how big it is and that sort of thing, and then kind of list the functions that that particular object has. And that object could be a molecule, it could be an atom, I suppose. It could be anything all the way up to the size of the entire biosphere, right? The entire planet, or I guess the universe, if you wanted to do it. We're going to be looking at molecule at the first level, and then kind of moving up through the biological hierarchy, we'll be looking at species next. So you can take a species that you are interested in and do the same analysis. Then the other aspects to these, once you've sort of described a noun, if you will, you have the how does it participate in the transformation of energy and matter? Everything ties in somehow to the transformation of energy, for sure, and matter, because things are made of matter. And so they're part of some material cycle. And so we're trying to figure out, like, where does that object fit in? Information flow, well, every object, there has to be some information that either it's contained in, or that codes for it, or that allows for it to be replicated, or whatever. And so that's kind of a different level of analysis. The evolution part is where you try to analyze, like, where did it come from? How did it evolve over time? What sort of factors influenced it? And we'll look at the phylogeny of that, and maybe some aspects of its history, and look at the mechanisms that influenced the evolution of that species, molecule, whatever it is. And finally, at the systems level, we'll look at, like, how does this tie into other potential systems? And so, like a species, when you do the analysis of that, it ties into an ecosystem or a community, right? So it immediately becomes a participant in a larger system. But within that organism, if we're looking at a species, it has organ systems that kind of interact, that participate in the creation of that phenomenon. So there's all kinds of ways to look at this. And at first, it might be completely overwhelming. So where do you get this information? Right. So you have these 5 categories and a phenomenon, and then you're like, okay, now where do I get it? So my first step is to go to some simple Google searches, just really simply. And the Wikipedia page for the phenomenon usually gives you a nice outline of these things, and you can probably pluck out many of the necessary components. I'm perfectly fine with you using Wikipedia for a general, content thing, if you like. Most of them are pretty accurate, although if you see something odd, you can always double check it. When you're searching in Google, your query, what you type in often gives you the results you either want or don't want. And so if I were doing serotonin, which will be my molecular example here, I have, instead of just typing serotonin, you might ask some very specific questions in the search field, right? So how did serotonin evolve, for example, if you didn't get that in the Wikipedia page? If you didn't get the structure, what is the structure of it? You can state things like, well, is there a relationship between serotonin and brain function? What is the phylogeny of serotonin? And maybe do an image search and see if you get the phylogeny of that. So being kind of specific, once you get down to the categories, sometimes helps in generating the info you need to put into your analysis. The sort of next level up would be to use something like PLOS or Google Scholar, where you're looking through peer-reviewed work. It's a little bit more complicated, but generally, if I'm looking up something like serotonin, or say a species, like, let's say, you took the species your team picked, in your team thing, and you decide, okay, I'm going to do that as my first species. You can type in the common name, or even better, the specific epithet, which is the Latin name of that species in Google Scholar, and it'll give you everything published on it. I wouldn't try to hit recent necessarily on like a species, because it may be that all the research was done, back in the 1970s even. And so then, so you'd want to make sure you can get some of the older references. But you get these scientific papers, and as you're aware, I'm sure by now, reading scientific papers is, it's an arduous task. It's a lot of work, and requires a lot of terminology and a lot of, all this stuff. It really makes them difficult. So what I would recommend when you're doing it for this assignment is that you skim the abstract, right? Just to kind of make sure that the article is, important to what you're looking for. Like, say, in this case, the evolution of serotonin. Maybe I find a paper with that in the title. So I'm like, oh, good. Then I'll skim the abstract to make sure it's talking about what I might want it to look to do. And then what I'll do is just scroll through the paper and look for figures that communicate to me, hopefully, what I'm looking for. When I did this assignment, probably just like you, I did it like last night. Anyways, and I found 9 different papers that I actually used, and I looked at many papers that I was like, oh no, that's nothing. But I had 9 scientific papers that I was like, oh yeah, these are useful. There's the phylogeny. Here's a picture of the genetic structure, etc. And so, and then finally, you can use any textbook that you like if you have a textbook for the class. You can skim through. When you have a weird phenomenon, you like serotonin, you can just look up in the index. Yeah. But like, let's say you had a species name. Your textbook is not likely to have a particular species as an example. So you might have to move back up the taxonomic hierarchy. For example, if you, let's say, there was one person that chose, I think, a toad, yeah, as an example for a species. And so if you're looking at toads, there may not be like a specific reference to the Pacific or Western toad in your textbook. However, it will have some information on a general category of amphibians, which is a class of chordates. So you might have to move up a little bit in the classification scheme to find info in something general like a textbook. So, just keep that in mind when you're kind of searching a textbook type thing. The Google and Google Scholar, they don't have that problem. You can type in whatever ridiculous esoteric thing you want, and you'll likely get something. But once again, if you find nothing about a species, you might back up a little bit. So if you're like, oh, I'm looking for this particular, someone I think posted something about like a pampas grass, and that's fine. And so if you're looking at it, but you don't find anything about that specific species, you can just look up grasses in general and see if there's information about the evolution and ecology of grasses in general. Anyway, so that's kind of where you go to get that info. And then just to give you an example, these, we'll move you guys over here for a second. So in general, I got these different figures from papers, right? So I'm looking up these info. These are the genes that participate in the creation of tryptophan, which is necessary before you make serotonin. And this is a set of genes that you'd find in E. coli. Okay. Then, and that was in one paper. And then in another paper, I found all of these different color-coded things over here are all the same or similar genes found in different species groups like cyanobacteria and yeast and fungi and et cetera, et cetera. And they're arranged slightly differently, and some are fused, et cetera. So you have this kind of general scheme, and then you have a whole bunch of different versions of that over here. And so I was thinking, well, what, for the information part, if I want to talk about the genes that create this thing, how do I summarize that? So this little sort of gray caption down here, this is literally from my notebook while I was writing things down. And basically, I just sort of sketched out a summary, very simple summary that has the different parts of this set of genes. A couple other genes that I found necessary, because these are all about tryptophan, and there's a couple other genes, the hydrolase and decarboxylase, that take tryptophan and make serotonin. And then the one at the bottom there, the monoamine oxidase, this is an enzyme that degrades it. So altogether, to make it and get rid of it, you need all of the genes that I've listed here, 10 different genes. I kind of looked up how big these genes are and then kind of added them together. So altogether, the 10 genes come out to about 20,000 base pairs. And this is the information, right, necessary to make and get rid of serotonin. The risk when you're looking up technical stuff is that you just cut and paste these parts and throw them all together and like, ta-da! And the trouble with that is that you probably won't understand what that means, right? If later I ask a question like, compare the information about this with the information flow about that, and if you've just cut and pasted pieces, maybe you don't really quite understand what they are, like, oh, these are different genes. I didn't know that, So it's useful to do some sort of a reduction, or, kind of, it's like putting it in your own words. For me, it's kind of putting it in my own diagrams that makes it useful and for me to understand. So I recommend doing some sort of a thing where you're manipulating this information in some way so that you really get a firm grasp of what it means. And don't worry, you don't have to do any of this stuff exactly like I've done. So let me kind of go through. And so here's the structure and function. Whoop. The first part of it, the structure part, for serotonin. And once again, this is right from my notebook. So when I was doing this, when I went to the Wikipedia page, I'm like, there's the molecule. So I drew the molecule, named it. It has another name. So I put that in there too, because you often see it referred to as 5-HT. I put in little details because it said, how big is it, et cetera. So I put the molecular formula and molecular weight and stuff in there. Yeah, so that happens. And then this is kind of lower on the same page, is the function. So, it turns out this molecule is found in all different kingdoms. So it's found in everything from bacteria to fungus to plants, animals, and it has slightly different functions in each group. So I kind of put down the ones that I thought were interesting. Now, when I'm trying to figure out, like, out of thousands of possible examples, what would I put here? Like, how do I decide? A couple things I try to do is pick things that might be useful later. And you were like, well, how the hell would I know that? and part of it is, are these functions that allow this to interact with other things, which will be useful later in systems and networks and things. In an ecological setting, are these roles that this can play and integrate into an ecological setting, which is also about interaction, like something eats it or it helps control something. Or finally, because we have this urban ecology theme, there's sort of a built-in sort of anthropocentric view. Like, are there useful things for humans, or does it interact with humans in some way? And so I put probably a little more about the role it plays in humans, the function, just because that's, that might be useful later in this class. Because hopefully your notes and these assignments you do will build, and you can use them later for different examples and certainly on exams. Anyways, one thing I noted is when I was doing this, for example, these various functions in humans, when you have a lot of serotonin, it improves your memory, dendritic branching, all these things. And then if you have too little serotonin, it leads to depression, neurodegeneration, decreased cognitive ability, all these things. So I realized that this was going to play on later. This is actually part of that systems, analysis. So you notice up here by function, you can't really see because it's from my notebook, the ink's a little dark, but I color code these things by the colors that were in the sort of original description. So if it's the blue is the form and function one, and then there's this sort of darker color here, which is representing, I guess, the purple from the possible interactions. I might use this stuff later. Anyway, so I kind of color code different things so I'd cross-reference them more easily. All right, so then moving on, in the, I guess the second category, which is kind of like flow of energy and stuff like that. First of all, I sketched out at the top here the general sort of flow of energy in biological systems. Whoop. Sorry, that might have been scary. Anyways, the energy flows from the sun, comes in as electromagnetic radiation captured by producers, plants, and other things that do photosynthesis. They're consumed by consumers, and then everybody gets decomposed, and the matter is recycled. The energy that comes in through the electromagnetic radiation is actually expelled as heat through this process. So the energy flows through the system and the matter is generally recycled. Now, that's the general pattern. The question is, how does serotonin fit into that? So I wrote down 2 notes here. Serotonin itself does not necessarily play a direct role, although because it's an internal communicator and it maintains the health and well-being of almost every type of organism, from plants to animals and fungi. They all make serotonin and use it as a signaling molecule. So without serotonin, they would not function. It's sort of like the whole system depends kind of upon serotonin. However, when I was looking it up, the precursor molecule, tryptophan, which you need to make serotonin, is a major component in photosystem 2. So it's very important in the photosynthesis, and therefore in the fundamental beginnings of the flow of energy in all biological systems. So even though it doesn't have a lot of details here, I tried to think about how this molecule and its associates tie in to this larger scheme. That's why I put that up there. Just to be kind of crazy, I sort of sketched out the the overall flow. This is all the biogeochemical cycles summarized, and I'll post this PowerPoint to the Canvas page if you want to see my crazy thing. Or even better yet, I suppose I could do a description of this in detail. The point of this right now is not to go over this diagram, but I kind of made this as part of this project because I wanted to see specifically if there were specific components or parts of this that this molecule played a role in. And so what I did was I took the basic molecules, chinops, right? You probably remember that from 10A, right? That's carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And then I looked up their biogeochemical cycles to see, like, how did they flow through the biosphere? And so you'll see them all here, right? All those different molecules. These, the nitrogen and CO2 and oxygen, these are all out in the atmosphere. The phosphorus, mostly it's a soluble thing. Rocks erode and then plants pick them up. The sulfur, comes from volcanism and other things, and then it moves into the biosphere, uptake by plants. It interacts a little bit in an anaerobic environment. That's hydrogen sulfide. But carbon, obviously, is probably the main thing you'd track. And it's taken up as photosynthesis, consumers, and decomposers through the process of either aerobic or anaerobic respiration. It ends up as CO2 coming back out. In the process, the energy moving through at all these steps is eventually ends up as low-grade heat. In the process of transferring it from electromagnetic radiation to low-grade heat, it does all this work. It generates the ability to do all this stuff. Anyways, so I sketched this out. Does this have to go in your notebook? No, not at all. I just wanted to point it out because depends on your molecule. your molecule might play a more important role in here somewhere. Like, I don't know what molecules you picked, but, you might want to think about a little breakdown like this to see how your molecule might participate. Later, when you do species, you will have to do one over the course of the semester from each sort of kingdom, whatever that means. But you'll note that plants, animals, fungi, bacteria, these things all are sort of different components or different compartments of this biosphere in general. So then they each participate slightly differently in the flow of energy and matter. So that this diagram might be more useful when you get to the species level stuff. Okay, and then if you just for kicks, if you wanted to, and this certainly does not have to go in your notebook. This is the flow of matter in the universe, right? So if energy flows from the sun through our system, you might ask, well, where did the sun come from? And so everything starts with nothing, and then there's a big bang and a molecular cloud, which then condenses and generates stars. Some of those stars will go through supernova cycles, be first, second, third generation stars. All of the elements are generated in these supernovae. So all the heavy elements, especially above iron, you have to go through supernovae. So all the stuff that's like the matter on our planet has to go through this process. And then all of this stuff eventually ends up sucked down a black hole somewhere and probably ends up back to nothing. And around it goes. Does this have to go in your notebook? No, this doesn't. I just wanted to do that for fun. Well, I guess it does make an interesting point. You might not have this problem, but I do. The internet is so full of interesting info, and my head's all full of crazy things that sometimes I get lost on tangents, and so this may happen to you as well. And sometimes you have to pause and think, oh, is this important? Is this important to the assignment? Am I wasting my time here? If I were doing this as a student in the class, I'd probably say this page probably is a waste of time. The one before it might have some use later, but you need to kind of be careful with that. Anyways, back to the point, I suppose, is the information flow. The simplest information flow for this molecule has to do with the information necessary to build it, the genes, right? The genetic system that develops it. And the genetic system, as you noted in a previous diagram, is a set of molecules called the trip operon, or this is for tryptophan. And the tryptophan pathway has a repressor and an operon and a bunch of different versions, genes in there necessary to make tryptophan. And then tryptophan at the end, if you make a lot of it, comes around and binds to the repressor and shuts off the system. To get from tryptophan to serotonin, you need a hydrolase and a decarboxylase. And to get rid of it, monoamine oxidase. So altogether, about 10 genes. You saw in that diagram previously, there's lots of variations of it, but this is the basic information you need. And that's kind of interesting in a funny way. 20,000 base pairs and all these genes is a lot of info. Yeah. The molecule is super simple, right? It's a little set of like two rings linked together. And all that simple molecule needs all of this information to generate it, which is kind of interesting, I think. Now, thinking about the molecule and its function, I came up with these other two diagrams. And here you'll note the two colors, the two dots, orange and purple. The orange is for the information flow part, but the purple, because I realized these are systems, they would, this kind of doubles for both, right? Both some systems it participates in and the information flow. But the molecule itself, serotonin, participates in the flow of information in your cognitive system, in your brain, yeah, and your body in general, but in your brain specifically. So your brain is all about processing information, right? It takes information from the environment, any change in the environment, change in temperature, touch, light, chemistry, all these things are transduced through your senses, filtered in your thalamus, then you perceive them, and then your brain does this thing where it integrates memory and objectives and whatever, makes a plan, and then you have an action, some kind of a thing. This could be a simple reflex arc, but it could also be some life planning thing. Anyways, if you have more serotonin, then this happens better, right? This is it improves this all the way around. Serotonin enhances dendritic branching and neuroplasticity, which facilitates this integration and planning. It enhances the dendritic branching, which improves the memory part of this. So serotonin, increases or improves this. And then I thought, well, if that's true, if you have a bunch of people who are serotonin enhanced, or people that are have lots of serotonin, then their social information flow will flow better. That whole social network will happen better. It will improve because people that are, say, suffer from depression or poor cognitive function or low memory, all of those things decrease your social information flow, which changes the society, changes the people, the whole social system you participate in. And so it's not just a matter of like what's happening in your brain, but then what's happening in the sort of social circles that you move in. And you might say, well, what does all that have to do with anything, particularly in this class? Well, I might note, and I'll mention this again, that one of the things we're looking at is the interaction between urban environments, which are enhanced, are by nature full of people, and by nature also have nature. So you have both, humans and nature interacting in these urban ecological environments that we'll be studying. Exposure to nature and access to nature improve serotonin dramatically. There are a number of scientific papers where they do basically just take you outside and put you in nature, or you play out in nature. And by doing so, it improves your serotonin, which then enhances your cognitive function. And then when you think about the social aspect of that, communities that have ready access to nature will thus be a higher functioning group than communities that are cut off from nature and don't have parks and aren't encouraged to go outside. So all of a sudden, there are all kinds of potential social justice issues and things that come up with regard to this. So it wasn't just a weird tangent, which I often go on to, but actually something that could apply to our class. Anyways, so moving on, the evolution of this system, what I did for this was I start, obviously I like to think in terms of diagrams. I might mention that, you probably noticed. And I like to sketch things, and that helps me. These, by the way, even though they're pretty crappy, are not, they're not my first draft. When I'm thinking, I sketch and take notes and draw. I can't really show you the table behind me, but I have a table full of pieces of paper with hundreds of little crappy sketches like this, in pencil that I erase and redo. And so in the end, this lovely sketch that you see in front of you is a culmination of many different thoughts and ideas. Anyhow, so I sketch for the evolution, usually the phylogeny. And in this case, because almost every organism on the planet has serotonin, the ability to make serotonin in some way or another. The pathway, those 10 genes at least that I mentioned before, have to be present in the last universal common ancestor. Luca is the last universal common ancestor of everything, had to have had that whole pathway. So that correlates to some process around 3.8 billion years ago, where the first cells start emerging. We have fossils of them. In that first condensation to these networks that were chemical and outside of cells, to condense them into cells where they were surrounded by membranes and began their autonomous lives. This whole genetic system had to be present because those same genes are found in everyone. Now, it could be that they're convergently evolved everywhere, but the genetic sequence is so common that it's very unlikely. So most likely it was found here. The only thing I note up here is that there's a loss of the tryptophan pathway in animals. Oddly enough, they maintain the serotonin pathway. That is, the couple of genes necessary to convert tryptophan to serotonin are still found in animals, but that's probably due to a genetic loss. I put a note up here that psilocybin is a unique convert that fungi developed. So they take tryptophan and convert it to serotonin and then make psilocybin, which is their own unique molecule. And I also made a little note, this little dotted line for me indicates an endosymbiotic event. That is the cyanobacteria that go on to become the chloroplasts and the plastids in these other eukaryotic cells. They contain all of the genetic information for this pathway. So probably it originated in cyanobacteria, which by chance happened to be the oldest fossils at 3.8 billion years ago. And when they were taken in as endosymbionts to become the plastids of these cells, they carried those genes with them. And so that's kind of interesting. Anyway, so that's my summary of that part of the evolution. The bottom part here, I listed some evolutionary mechanisms that I kind of skimmed and read about. For example, that set of TRP genes that you saw, it starts as a single gene, and then that gene's duplicated and then mutates separately to become slightly different versions. Yeah. So gene duplication is often how you make a set of very similar genes. Hox genes in animals, for example, evolved in that same way. It's a very common genetic mechanism to generate copies and then have them diverge to make genetic diversity. Gene rearrangement. I don't know if you remember that diagram earlier. On the right, there was a column of a bunch of fancy colored gene sequences, all different colors and things, looked like a little rainbow. And those were all genetic rearrangements of the same set of genes. So in different groups, whether it was E. coli or in a fungus down below or whatever, all of the different groups have slightly different rearrangements. So the gene sequence has been rearranged. Endosymbiosis, pretty important for this group of genes because of this plastid development thing where the endosymbiotic event creates the plastid, but then carries the genes along with that. There are examples of horizontal gene transfer, and horizontal gene transfer is when it often happens in bacteria most commonly, but also between bacteria and eukaryotic hosts, where a gene from the bacteria is transferred. Usually it goes that direction, sometimes the other way. So there are mechanisms for gene transfer across different species, in fact, across different kingdoms often, that happen because of these sort of endosymbiotic events, or with bacteria, that's just how they take up genetic information from the environment. Anyways, and then of course the traditional mechanisms like mutation, natural selection, and genetic drift all play a role. And then I found a weird example recently, and they haven't even named this. Probably it's a new, I don't know, phylum of protists over here. But this kind of thing, the example was called kleptoplastic endosymbiosis, which makes sense. Kleptoplastic means you're stealing the plastid, in this case, like a chloroplast. And the example was a dinoflagellate, which is a eukaryotic cell, a type of protist, takes in an entire diatom, which is another eukaryotic cell. So the eukaryotic cell becomes essentially an organelle inside another eukaryote. This would be way down the line in endosymbiosis. It's not primary, secondary, not even tertiary. This is like way down the endosymbiosis line. And that obviously all the genes from the diatom go into the dinoflagellate. So now this new thing has two copies of the whole suite of genes that are slightly different. And then in this case, it also took in another bacteria and made another plastid with a whole different suite of the same genes. So it has three sets of genes to make serotonin, all of them different. So, so it's a crazy eukaryote. They don't even know what to name it. the one paper said they're going to call them dinotoms, which is kind of a cool name or whatever. But anyways, it's kind of interesting. It demonstrates a couple things. One, how these gene sets move around, how this molecule has been shared and diversified over time. Because goodness knows, the dinotom now can make 3 versions of serotonin. Who knows what that means in terms of its biology? But it also kind of elaborates and makes a point that evolution at the phylum level can happen instantly. Yeah, you make new phylum in one event, right? A single event, a single endosymbiotic event that happens literally in seconds can lead to an entirely new phylum. Whereas species might take a little longer to evolve over some of the more traditional mechanisms. So evolution, it kind of demonstrates this weird pace of evolution that is unexpected. Okay, so anyway, so that's all the notes I had for the evolution of this. Once again, all this talking I did explained what this means to me. When you make a notebook and you're like, oh, I do this sketch in here, but you don't have a lot of notes, I can look at it and maybe I don't understand what you meant by this diagram, because you didn't do a long explanation like I just did. So I might ask you, yeah, when you turn this stuff in and you put in a sketch like this, if I'm not sure what it means, then I'll probably, put a comment like, hey, could you explain your evolution part of this to me? And so there might be a little dialogue, but I want this to be to be very personal in a way, right? I want you to make it your own, however that looks, and then I'll try to figure it out from there. A little explanation here and there wouldn't hurt to help me through what your little coding might be or whatever you might do. Anyways, so moving on, now we make it down to the systems level. And the first system this thing participates in that I hadn't put up before was the molecular network. That is the molecules that are formed in those pathways to create tryptophan and serotonin. And you can see that my spelling's not that great. There's no E at the end of tryptophan. Anyhow, these are my notes and that's just the way it is. So anyways, I put down some stuff that I think is useful in this like molecular network. Because if you remember, when we were looking at the hierarchy of biological systems, you had sort of, particles, atoms, molecules, and the next thing is molecular networks. And molecules obviously never exist by themselves. They're always part of a process, a network. And so its immediate network would be the things that lead up to making it and the things that it decomposes into. So anyways, this is the sort of sketch. And I put a few of the interesting functions in there. So tryptophan by itself goes into photosynthesis by being part of photosystem 2, main part of it. And yeah, so then when you run the hydrolase to start to convert tryptophan to serotonin, you have to add oxygen. That's where that little OH group comes from. And so it's kind of interesting. And then the decarboxylase, you take off one of the carbons. And it's interesting that photosynthesis is all about creating oxygen and taking up CO2. So it's kind of interesting that the process of moving tryptophan to serotonin actually kind of mimics or connects to that process of photosynthesis. I found that interesting, so I put it in there. The other thing is serotonin isn't sort of it's a very important molecule for humans and for animals and for everything in terms of signaling, but then it is also part of a pathway that connects to some other very important molecules. Auxin in plants is like a major growth hormone that sort of is used for plants to grow, to elongate stems and root tips, for them to adjust to gravity. When a plant leans over, it brings it back up. When light hits one side, the plant grows a different way. And all of that is conducted by auxin. So it's a very important plant hormone. Melatonin in animals, along with serotonin, pretty important for our cognition and brain function. Melatonin helps us with sleep, for example. And so it kind of makes sense, right? When you think about the overall role of serotonin being sort of cognitive well-being. Obviously, it then also enhances sleep by directly making melatonin, which enhances your sleep-awake cycle. And then finally, serotonin in fungi is converted to psilocybin, which is used for defense in the mushrooms and things that make it. It's a way to prevent animals from eating it. But but now it's being looked at as used for medicine and things like that. Anyway, so that's the first level. And then I kind of sketched out some stuff where how it participates in the animal physiological system, right? Once again, there's this kind of hits on multiple parts of that 5-part system. And I just took some notes here about, the different parts. For instance, it's made in the Rafe nucleus in the brain. The platelets make it in the cardiovascular system, and 90% of our serotonin is actually made in the enterochromaffin cells in the digestive system. That was interesting to me. And I learned that the microbiome produces molecules which help facilitate our neuroprotection. So it facilitates our cognitive function and stuff through a molecule also in the tryptophan pathway. So it turns out then your diet, which influences your microbiome, can influence your cognitive ability through this same system. So that's pretty interesting. Kind of want to avoid junk food then, right? That's only if you want to be smart. Anyhow, then these are some of the receptors. It turns out that serotonin binds to all different types of receptors. And in doing so, it does different functions in different cell types. So while the molecule is the same, in these different physiological systems, it operates differently because it uses different receptors, which trigger different responses in the cells. And finally, I noted the 2 terminating methods, the ways to get rid of serotonin. So you make serotonin and then you have to get rid of it. Monoamine oxidase is an enzyme that breaks it down. And the serotonin transporters, which transport it back to be broken down back into cells, it turns out those 2 are the main pharmacological mediators. So there are drugs made which inhibit the monoamine oxidase, so you keep higher levels of serotonin, and SSRIs, which prevent the reuptake of serotonin, keep more serotonin in the synapse. So these are our currently two pharmaceutical ways of intervening in this system to increase serotonin to avoid things like depression and some of the other diseases associated. And that's that. So let me ask you if you have any questions about that. I'm sure you must.
So for future notebooks, like assignments like the BioCORE, this is kind of the layout you want it to actually be, and not more of a, oh, I'm actually writing stuff down on Word. You actually want us to just like legitimately just sketch our ideas or what comes to our mind more than actual definitions and facts.
Okay. Yes and no. It really depends on your process. I wanted to demonstrate this like authentically the way I do it, right? So I do this way. This is exactly what I wrote down. This is actually, like I said, kind of a second draft. I have like 1,000 pieces of paper. Like I look at the Wikipedia page, and usually I keep notes, in little categories or whatever. And then I compile them. Like, what are the main points I learned? Now, if your process is different, your process might be to open a Word doc, and as you're looking stuff up, type stuff in, even cut and paste stuff over to your Word doc. So you're like summarizing your research digitally in an immediate way. Other people use different digital, different apps for the same idea. So it might be that yours is a Word doc and it's all done sort of typed and stuff like that. And that's fine too. So they can be very diverse in what you make. I don't know if that helps, but it's okay. The content's important, that you have some ideas of your own. I don't want to see like a Word doc that's just cut and pasted stuff, because then I don't know that you understood it. Yeah. it has to show some manipulation or some, overall synthesis that puts them together. And don't worry, I'll give you feedback on the first one for that.
Got you. Yeah, that makes a lot more sense now after you're doing it. It's like you're not really looking for a specific structure, you're just looking for a thought process.
Exactly. Yeah. So other questions?
Can you go over what the systems is supposed to cover? Because I didn't quite understand that when I was doing the assignment.
Sure. Yeah. That's it. Okay. So with a molecule, it really depends upon like where it participates. For example, in this, where you can see the animal physiological systems, it participates mainly, it's produced in 3 of our physiological systems. But then you're like, yeah, but then you had like cognitive system, and then there was this social system, and then, so where does it fit? What do you have to do? And it kind of depends, once again, upon your research. The idea of the social system as part, the cognitive system participating in the social system, that was sort of an idea I had. I didn't look that up anywhere. It just came to me that, oh yeah, that's got to be true. As I tried to tie this idea into our class project, where we're looking at social justice and access to nature and stuff. The when you get to the animal or plant or the species, that's the next level we're going to do. That has a little more defined set of systems, right? Molecules really hard to say, but and like say you took an animal, like I don't know, a toad. So you have this toad, and you're like, okay, what systems does this toad participate in? Well, first of all, the toad has its own organ systems inside it that makes it. Yeah. So there's a set of systems that comprise the toad. So you would say, okay, I'll look at its organ systems, just kind of lay those out, how they interact to make this toad. And then this toad participates in a local community. It eats things eat it. It uses other species in different ways. It's connected to its local ecology. And that's the second system it participates in. And if you just did that, the systems that make it and the systems it participates in, you're good. You don't have to do anything beyond that. The other things that I was talking about are just if you happen to think about how it might connect to other systems. For example, I suppose, let's say you chose a mosquito, right? And so your mosquito, now it has organ systems that participate in making it. So that's part of it. But then it participates in the local ecology, part of it. Then one more system, many of the mosquitoes, and especially around here, carry various diseases, right? And so then it interacts with sort of a human ecology, and through our, through the diseases that it might carry. So, and so it kind of depends on the species you choose as to what systems it participates in. And if that seems all very vague, that's okay. It's supposed to be, and it's kind of up to your exploration and your own ideas to how you might connect things. I don't know if that answered your question at all.
No, it did for a species, but how about for like a molecule or something, like an example you gave?
Okay.
Like, because we have to do both a molecule and the species, right?
Okay. Give me an example of a molecule and I'll try to do that. Go ahead. Just anyone, just whatever molecule you choose.
So when I was doing the assignment, I was looking up luciferin. It's like that thing responsible for bioluminescence. So maybe something like that.
Okay. All right. Wow. Okay. Luciferin. So luciferin. And all right. So it has a molecular system that it's connected to. So zooming in at system 1 level would be the molecule, the steps that make it, and the way you decompose it or whatever. So with luciferin, I remember there's an enzyme luciferase, for example, that's part of the process. And then there must be molecules that are used, or maybe a gene in this case, that codes for luciferase, right? and the steps. So there's like a molecular network, and that would look, let's see, oh yeah, that would look something like this. Yeah, the molecules to make it, and and how you degrade it. And there might be these are molecules, and I noted the enzymes here, right? So in your case, you would have luciferase, but then there might be some others participating in this. So one network, molecules. The next level up is, in the case you've shown, is a sort of ecological network. Yeah, it's for instance in planktonic systems, where the organisms that produce luciferin and use it to like flash light, it's a predator deterrent. Yeah, so predators come in to eat it, it flashes the light, and it scares off the predator. and so then it's like part of an ecological network, right, as a defense molecule against predation. So there's a network of ecology there that's kind of going into that. And then finally, in a very weird way, as you probably noted, maybe this is where you heard about it, is that molecule is used in research, right? for a lot of things. We use it as a signaling molecule that the genes have been transferred, all kinds of stuff. So all of a sudden, that molecule is part of a sort of social scientific network where it participates as from that organism, maybe the dinoflagellate that produced it. We've taken the gene, we now have put it in mice and all kinds of other things to make them glow in the dark. I think there's even cats that glow in the dark now. And so now it's become part of this bizarre genetic engineering practice and our research practices. So you could kind of sketch out kind of a weird network of how that's participating. So right off the top of my head, I would do the molecular network, the ecological network, and this weird genetic engineering research thing that's going on. So I don't know. That's right off the top of my head. That's what I would do with that one. Does that make sense?
Yeah. So, okay. So it's like the molecular network, and then I guess you're supposed to branch out to see how like this molecule is used in like certain like ecologies, whatever, with like certain species. And then you were saying that even like we use it for research, so we even go like that far into it, or I guess that deep into the level of like what this network is.
Yeah, and that stage, if you will, is useful for the class because we're doing this sort of urban ecology theme, which is really about the relationship between humans and nature. Yeah, right. I mean, we create the urban environment, et cetera. So any link you can make to human social systems, or, I mean, some things people eat, some things, people use as tools, people grow things in their garden. All of these human things are part of the network that these different phenomenon integrate into. So this molecule just happens to be a huge research tool. So it's kind of an important thing that way. If you missed that part, if you didn't include it, that would be okay. If you had the molecular network and the ecological network and you did that, that's fine.
Oh, okay. So really we just need the molecular and ecological network.
Yeah, I mean, those are the like natural things that molecule participates in. Yeah.
Okay, thank you.
Yeah.
I have a question, Professor.
Sure.
Would social systems also be defined as an organism's or molecules' symbiotic relationship with other molecules and organisms?
Yes. Right. Yeah, so you're talking about molecules, right?
Or organisms, either or.
Sure. Oh, right. Yeah, of course. So when you're looking at a species or any organism, participates in a local ecology, and when you draw arrows connecting that species to others, the arrows can will often, the first step is to go, oh, what eats it? What does it eat? So you make some arrows there. But then there are all kinds of other things that they do, right? They interact in many different ways. For example, if you put oak tree in your set, if that was your species, then oak trees are like a house, a home, an architecture that other species use to nest in and hide in and do other things. And so there become all kinds of, possible connections. And then when you connect that out to humans, let's say you're connecting oak trees to humans, you may notice that oak trees are, well, I mean, they're people like oak trees. So we have this weird association with them. We don't eat them. We don't live in oak trees, but we protect them and we grow them. And so we're connected directly to them in some strange way. So yeah, you're looking at a broader set of connections. Did that answer your question or confirm your idea?
Yeah, that did. Thank you so much.
Okay. Just as a quick aside, I didn't put it in this show, but I had this other thing that for the serotonin that I might have put in my notebook because it links to our social justice equity thing and the idea of urban ecology. I looked up a couple of different papers that indicated that access to nature and sort of nature therapy was a really big deal, and that lack of access to nature led to communities with higher levels of depression and suicide. And so I made a little diagram that shows the connection between nature, serotonin, and all of these neurological functions. And so, just to remind myself to connect that later. And then there were a bunch of recent papers that I didn't realize. And I didn't go too far into this, but there's a thing called the psychedelic renaissance. Apparently, there's a whole new wave of research and like 11 new companies that are just starting up. I don't even know how they're doing it because these psychedelic drugs are illegal, but they're doing research and showing that it improves the serotonin system and enhances all of this stuff in ways that they had not ever expected. One article I read was a company that's developing a psychedelic therapy for vets with PTSD, and it's fixing that. And then they couple that with access to nature. So like walking in nature, eating mushrooms, whatever, it really enhances their well-being. And it only takes 1 or 2 exposures to treat entire diseases. Anyway, so I threw in a couple other connections that I thought might be useful later in my notebook. And that might be useful for you as well. Okay, so are we done with all that? Any other questions about the BioCORE example and stuff? Feel like you could give it a try now?